And actually it turns out that you could probably, oh, repeat the question. Sorry. I'm a slow learner. So the question was, are there topological similarities between proteins which tend to form these amyloid type, these aggregated structures, or, and to comment on that. And what's fascinating is that there's increasing evidence that any protein can form one of these aggregates. And actually, my favorite protein, it was the protein of my graduate study, so it's sort of, I don't know, like you fall in love with a protein at some point in time in your life. And the protein I fell in love with was myoglobin, which is the protein that holds oxygen in your muscle. In many ways, the most boring protein on the planet, but it's the one that I studied, so it's the protein that I love. Myoglobin it's alpha helical. It is, a pretty dull protein. It's not involved in anything terribly interesting. Under the right conditions, you can make myoglobin form amyloid fibers, which look exactly like the crystalline fibers or the Alzheimer A beta fibers. So it appears that this is a general phenomenon that any protein can do. There's certainly only some proteins that actually exhibit this behavior regularly in vivo that we identify, so that seem to be biologically significant. But from a chemical standpoint, it's a general property of the proteins. And in fact, there have been a few cases made that, that we can really think of successful folding of a protein as being a challenge of, the main challenge in successful folding of a protein is preventing the unsuccessful aggregation of a protein. And that in vivo, there's some competition between all proteins ending up in the aggregated state versus ending up in the stable state. And as people started studying the cell biology of this more often, and this is getting into an area that is not my exact area of expertise, but I think it's fascinating work. It turns out your cells have an awful lot of machinery that's out there to try to vacuum up misfolded proteins, because if you think about it, even if it only happens once in a while, you're making proteins so often that you're going to have some proteins which don't fold correctly. And it turns out healthy cells, have a means of trying to vacuum up and deal with and identify misfolded proteins and get rid of them before they cause trouble. And so there's some interesting argument that something like Alzheimer's disease, the issue might not be the protein problem in terms of the protein forming this state, but a problem going on in the cellular mechanism that is breaking down and preventing the vacuuming up of this protein, misfolded protein, that eventually leads to the identification of the aggregate. So it's sort of, I think we're in the midst of a sort of change in perspective in terms of it may not be the molecule that's most important. It actually may be the cell and the way the cell responds to the misfolded proteins that's actually important. And that in certain, whether it's a genetic effect or an environmental trigger, but in some cases, the cells stop responding efficiently to try to get rid of misfolded protein, and that leads to the disease state. Does that? Yeah.
Where do you go from here?
Where do you go from here?
You can repeat that.
Yes. So where do I go from here? So we've actually been working on a couple of different things. One is, can we begin to think about designing molecules which can prevent aggregation? So molecules that might be able to bind to proteins and prevent them from sticking together. And if so, could we design molecules that could actually be effective in some way in treatment of some of these diseases? Cataract, I think, is an interesting case because they're really the only treatment for cataracts is actually a surgical treatment now. And I think it's not one way to put it. It's not the worst surgery imaginable, and certainly in Western, well-developed societies, it's become, I think cataract surgery is now basically outpatient and is, relatively routine. But cataract is the leading cause of blindness worldwide, and that's in part because in most of the world, cataract cataract surgery isn't really accessible and is far from being outpatient and routine. So, say the development of something which could be applied as a topical eye drop, for example, would be attractive in the sense of really actually making treatment for cataract far more accessible to worldwide populations. I mean, in addition, it would make life better for Americans too. But it would, in parts of the world where surgery is really not a practical option right now, that would actually be a significant breakthrough. So in the larger picture in this field, understanding more about these molecular details might allow the design of molecules which could break up and prevent the aggregation, which might be able to be applied to the eye in ways that are effective and low-cost treatments.
This is undergraduate educational research.
Right.
There are big companies doing work in this area, I assume. I'm not in that area. But if you need funding for this kind of educational research, do you apply to the research side or the educational side for that educational research?
That's a good question. So the question was, basically this is work that I'm doing in a context really of as an educational project, but it's also research that others are engaged in a from a pure research perspective. And where is funding support for this come from, or what side does funding support for this come from? And there's been a change in our major national funding agencies. So both the NSF and the NIH, the National Science Foundation and National Institutes of Health, in beginning to recognize that there is, it's a significant national problem not only to produce interesting research, but also to train the next generation of scientists. And that could we do those things at the same time? And within the National Science Foundation, for example, demonstrating that research is going to have a broader societal impact, including actually the education and training of students and the next generation of scientists, is an important criterion in the judging the funding of an award. And so, if you can put together an effective research program that is doing productive work, but also is training students, then it's possible actually to get funding for work through that mechanism. That said, federal funding for research is always in a very fragile state of things, and especially in recent years, has become increasingly fragile. And programs that are training students are some of the ones which have been most likely to be on the danger block or the cutting board for things. From a policy perspective, though, I think that approach is right on target. That there should be a niche for research that is effective at training students and ensuring that we have a well-educated workforce and a well-trained citizenry in terms of scientific problem solving. That also is actually good, solid science as well. So I don't know if that kind of answers, but